Dear friends, once again it's hearty welcome in this new lecture. Dear friends, in our today's lecture we are going to study about mechanism of crossing over and then the topic sex linkage, types of sex linkage. Actually friends, you people have studied about crossing over in your 11th class, but then and then here we are going to discuss about the crossing over. So let us start. Crossing over, this process has great importance because crossing over is responsible for the development of genetic variations, genetic recombinations in the population. You people have studied in 11th that in sexually reproducing animals, at the time of gametogenesis, at the time of gamete formation, it occurs the meiosis, that is meiotic division. We know that meiosis, it is a process in which the diploid parent cell divides to form 4 haploid daughter cells. And that's why the meiosis or meiotic division is also known as reduction division. Now before going to the crossing over, let us recall briefly about the meiosis. As I said just now, that meiosis, it is a type of cell division during which the diploid parent cell divides to form 4 haploid daughter cells. It means during meiosis the chromosomal number reduces to half, and that's why the meiosis is also known as reduction division. इयत्ता 11वी मध्ये आपण शिकलो आहोत की meiosis हे एक प्रकारचं सेल डिव्हिजन आहे आणि सेक्शुअली रिप्रोड्युसिंग ऑर्गॅनिझम्स मध्ये गॅमेट्स तयार होत असताना, गॅमेटोजेनेसिस होत असताना meiosis ही क्रिया घडून येते, किंबहुना because of meiosis from the diploid germ mother cells haploid gametes are formed. Now we know that the meiosis, this process takes place in 2 successive divisions such as meiosis first and meiosis second. We know that the meiosis first is the true reduction division. It is true reduction division because during meiosis first the diploid parent cell divides to form the haploid daughter cells, 2 haploid daughter cells, and in meiosis second that 2 haploid cells formed at the end of meiosis first further divides mitotically, that is equational division, to form 4 haploid daughter cells. Now the meiosis first, it involves as usual karyokinesis, that is division of nucleus, followed by cytokinesis, that is division of cytoplasm. And we know that karyokinesis occurs in 4 steps such as prophase, metaphase, anaphase and telophase. Now in meiosis first, the prophase has again substages. The prophase has again substages such as the leptotene, zygotene, pachytene, diplotene and dikinesis. And we have studied that the actual process of crossing over takes place in the pachytene substage of prophase of meiosis first. And here we people have to study the mechanism of crossing over. Initially, as I said, this crossing over process has a great importance because it brings about the genetic variations in the population, because crossing over is responsible for genetic recombination. It is responsible for the genetic reshuffling and it leads to the development of variations in the population. क्रॉसिंग ओव्हर या प्रोसेसला अतिशय महत्त्व आहे, कारण क्रॉसिंग ओव्हरमुळे त्या ठिकाणी जेनेटिक रिकॉम्बिनेशन घडून येतं, जेनेटिक मटेरियल मध्ये रिशफलिंग घडून येते, रिअरेंजमेंट घडून येते आणि पर्यायाने त्यामुळे नवीन अपत्यांमध्ये ऑफस्प्रिंग्स मध्ये व्हेरिएशन्स निर्माण होतात. यू नो व्हेरिएशन्स मीन्स डेव्हलपमेंट ऑफ सर्टन न्यू कॅरेक्टर्स. पॉप्युलेशन मध्ये नवीन कॅरेक्टर्स दिसायला लागतात and it supports, you know, the variations support the process of evolution. आणि व्हेरिएशन्स मुळे सजीवांमध्ये नवनवीन गुणधर्म निर्माण झाल्यामुळे इव्होल्यूशन उत्क्रांती या प्रक्रियेला त्या ठिकाणी सपोर्ट मिळत असतो. नाउ द क्रॉसिंग ओव्हर, द मेकॅनिझम ऑफ क्रॉसिंग ओव्हर, प्रोसेस ऑफ क्रॉसिंग ओव्हर इज बेस्ड अपॉन द ब्रेकिंग and rejoining theory proposed by Darlington. It was one scientist, Darlington, and he proposed one theory, that is breaking and reunion theory. Actually, the two major scientists, one was Belgium cytologist, namely Janssens, and another great geneticist, T.H. Morgan, contributed a lot in the study and discovery of mechanism of crossing over. क्रॉसिंग ओव्हर कसं घडून येतं, ही प्रक्रिया कशी घडून येते, हाऊ इज द मेकॅनिझम ऑफ क्रॉसिंग ओव्हर, इट वॉज स्टडीड वेल बाय T.H. Morgan and before to that little bit contributed by Belgium cytologist Janssen. So now let us talk first of all the definition of crossing over and then the actual process of crossing over. Let me define what is crossing over. Crossing over, it is a process in which it occurs mutual exchange of corresponding segments between the non-sister chromatids of homologous chromosomes. I am going to repeat the definition of crossing over. It is the process of mutual exchange of corresponding segments between non-sister chromatids of homologous chromosome during the Pachytene substage of prophase of meiosis first. Here I am going to write briefly the definition. Crossing over, it is mutual exchange of corresponding segments between non-sister chromatids of homologous chromosomes or homologs. Now let me explain you actual mechanism. Crossing over, it involves the 4 steps. First step, synapsis. The pairing of homologous chromosome along their length is simply known as synapsis. All of you are known in diploid cell there are presence of 2 sets of chromosome and always each chromosome is in the form of pair, and in synapsis it occurs simply the pairing of homologous chromosome due to the synaptic forces. Due to the synaptic forces, the homologous chromosomes, structurally and genetically identical chromosomes, are attracted towards each other and they lie close to each other along their length. This simply process of pairing of homologous chromosome is known as synapsis. Let me show in diagram. Consider here is supposed to be the cell and nucleus. Just consider here I am going to show 2 chromosomes. Now here these 2 chromosomes are supposed to be homologous chromosomes. आपण असं कल्पना करूया की या सेलच्या न्यूक्लियस मध्ये अशा प्रकारे ही एक होमोलॉगस क्रोमोझोमची जोडी आहे. नाउ I said synapsis, it is simply the pairing of homologous chromosome. The homologous chromosomes are attracted towards each other. Homologous chromosome परस्परांकडे आकर्षित होता due to certain synaptic forces and they lie close to each other along their length. Let me show you. Here is homologous chromosomes. Now, in this phase, their chromatids are not visible. In this phase, their chromatids are not visible. You know chromatids, these are the components of each chromosome. So, in this first step, only a pair of homologous chromosome is visible, and such pair of homologous chromosomes is also known as bivalent or may be known as dyad. Now come to the second step. The synapsis, it takes place in zygote phase, zygote substage of prophase of meiosis first. Now second step is tetrad formation. Tetrad formation. The chromatids of each chromosome, slightly separate from each other. The chromatids of each chromosome slightly separate from each other, and hence now in all 4 chromatids are visible. 4 chromatids are visible. 2 chromatids of each chromosome. Let me show. Suppose these are 2 sister chromatids of one chromosome. We know that chromatids always remain held together by means of centromere, and here are 2 sister chromatids of another homologous chromosome. It means here are formation of 4 homologous chromatids, and this group of 4 homologous chromatids is also known as tetrad. The tetrad formation takes place in pachytene phase of prophase of meiosis first. Next step is actual crossing over. Or here in crossing over, it occurs the mutual exchange of corresponding segments between the adjacent non-sister chromatids. Let me explain. क्रॉसिंग ओव्हर मध्ये प्रत्यक्ष नॉन सिस्टर क्रोमॅटिड्स जे आहेत, ऍडजेसंट नॉन सिस्टर क्रोमॅटिड जे आहेत, त्यांच्या कॉरस्पॉन्डिंग सेगमेंट्सचं एक्सचेंज होतं, म्युच्युअल एक्सचेंज होतं, समप्रमाणात देवाणघेवाण होते. नाउ प्लीज लुक ॲट दिस डायग्राम. इन दिस डायग्राम you can notice these are the 2 chromatids of one chromosome and these are 2 chromatids of another chromosome. So obviously these are sister chromatids of each other, these are sister chromatids of each other, but this chromatid and this chromatid, these are non-sister chromatids. So we can say the adjacent non-sister chromatid, adjacent याचा अर्थ एकमेकाच्या जवळ असणारे जे नॉन सिस्टर क्रोमॅटिड्स आहेत, they coil, twist around each other at one or many places. They twist or coil around each other. Let me show here. At one or many places. Look, the adjacent non-sister chromatids, they twist, coil around each other at one or many places. Actually, this is nothing but the crossing over, and this point of crossing over between the two non-sister chromatids is also known as chiasmata. It is known as the chiasmata. Now, at the point of crossing over, that is at the chiasmata, that chromatids break off. Chromatids break off by the action of enzyme restriction endonuclease. Restriction endonuclease या एन्झाईमच्या इन्फ्लुएन्समुळे हे chromatids break होतात at the point of crossing over, that is at the chiasmata, and again they rejoin with the opposite chromatid by the influence of enzyme DNA ligase. DNA ligase. So actually here it occurs the exchange of corresponding segments between the non-sister chromatids. Now this exchange is so precise, so perfect, that there is either no loss of even a single gene. There is no loss of even a single gene of any of the chromatids. After crossing over, the last step comes, that is nothing but the terminalization. Terminalization, or it may be called as disjunction. Terminalization or disjunction. Of course, the tetrad formation and actual crossing over takes place in achating substage or prophase of meiosis first, and then these two homologous chromosomes, they tries to go away from each other because the synaptic force ends, and thus the homologous chromosome tries to go away from each other. They tries to go away from each other. This is known as the terminalization or disjunction. It takes place in the diplotin and dikinesis. Of course, the terminalization not get completed in prophase. It continues in metaphase also. So, in this manner, it occurs the mutual exchange of corresponding segments between the non-sister chromatids of homologous chromosome. And now, due to crossing over, out of these 4 chromatids, the 2 chromatids show genetic recombination. आता या ठिकाणी क्रॉसिंग ओव्हर झाल्यामुळे एकूण जे 4 क्रोमॅटिड्स आहेत त्यापैकी हे 2 क्रोमॅटिड्स की ज्यांच्यामध्ये प्रत्यक्ष क्रॉसिंग ओव्हर घडून आलं त्यांच्यात जेनेटिक रिकॉम्बिनेशन दिसतं आणि म्हणून त्यांना म्हटलं जातं क्रॉस ओव्हर्स. These chromatids are called as the crossovers or recombinants, and remaining two chromatids which has not actually involved in crossing over are called as the non-crossovers or non-recombinants. Here we have completed the study of mechanism of crossing over. Now we people have to study about the sex linkage. Dear friends, actually we have studied about the linkage in our previous lecture. You know that the linkage is the tendency shown by the genes present on the same chromosome to remain together during hereditary transmission during inheritance. लिंकेजचा अर्थ काय होतो हे आधीच आपण शिकलेलो आहोत. Now here we have to study about the sex linkage. First of all, let me tell you the meaning of sex linked genes. Very simple. The genes which are present on the sex chromosome and are responsible to control general body characters except sex are called as sex linked genes. Let me elaborate and let me explain you properly. all of you are known, in human being, two types of sex chromosomes are present, such as X and Y. Now, these X and Y are known as sex chromosomes because these chromosomes are responsible for the determination of sex of that individual, isn't it? It means that the X and Y chromosome contain the genes for sex determination. याचा अर्थ X आणि Y या chromosomes वर sex determination साठी genes present असतात. But besides that sex determining gene, the sex chromosome also contain some additional genes which are responsible to control other body characters, general body characters, and has nothing to do with the sex. Such additional genes present on sex chromosome controlling different body characters are known as sex-linked genes. Is that clear? याचा अर्थ सेक्स क्रोमोझोम वर सेक्स डिटरमाइन करणारे जीन्स तर असतातच, पण या सेक्स डिटरमाइनिंग जीन्स पेक्षा आणखीन काही अतिरिक्त ॲडिशनल जीन्स पण सेक्स क्रोमोझोम वर प्रेझेंट असतात की जे इतर जनरल बॉडी कॅरेक्टर्स नियंत्रित करतात. त्यांचा सेक्स डिटर्मिनेशनशी काही संबंध नसतो. अशा जीन्सला म्हटलं जातं सेक्स लिंक्ड जीन्स. ब्रीफली once again, sex linked genes are the genes present on sex chromosome and are responsible to control general body characters. And the characters or traits controlled by sex linked genes are called as sex linked characters. The characters or traits controlled by sex linked genes are known as sex linked characters. या sex linked genes मार्फत जे जनरल बॉडी कॅरेक्टर्स नियंत्रित केले जातात त्यांना म्हटलं जातं सेक्स लिंक्ड कॅरेक्टर. And obviously the transmission of such sex linked characters from parents to the progeny, or we can say from one generation to the another generation, is known as sex linked inheritance. Sex linked inheritance means the inheritance or transmission of sex linked characters from parents to their progeny. And this entire phenomenon of transmission of sex linked characters is known as the sex linkage. Actually, we people have to study about the types of sex linked genes, as there are two types of sex chromosomes in human being, such as X and Y. So there are two types of sex linked genes also, such as X linked genes and Y linked genes. Means the genes present on X linked, and genes present on Y linked, about which we have to study in detail later on in our next topic. Now let us talk about the types of sex linkage. There are two types. First, complete sex linkage. Complete sex linkage and incomplete sex linkage. Incomplete sex linkage. Actually, we people have studied the meaning of term complete linkage, incomplete linkage. Okay, but here the same concept and same definition is implemented, only concerned with the sex linked genes. So let us talk about the complete sex linkage. Complete sex linkage is exhibited by the genes present on non-homologous region of sex chromosome. Listen carefully. Complete sex linkage is exhibited, complete sex linkage is shown by the genes present on non-homologous region of sex chromosomes. We have studied about the structure of sex chromosome. Suppose it is a X chromosome, Y chromosome, we know that the X chromosome is longer, submetacentric, Y chromosome is shorter, acrocentric, and I told you that some part of X and Y chromosomes does not show identical genes, and therefore such parts are known as non-homologous regions. Non-homologous. While some of the part shows identical genes, therefore it is known as homologous region, homologous part. Now I said the complete sex linkage is exhibited by the genes present on the non-homologous regions of X and Y chromosome. जे जीन्स X आणि Y क्रोमोझोमच्या या नॉन होमोलॉगस पार्टवर असतात ते कंप्लीट सेक्स लिंकेज दाखवतात. म्हणजे काय? इट मीन्स these genes present on non-homologous part, they remain together and inherit together generation to generations without separation. They cannot be separated from each other. अर्थात या X क्रोमोझोमच्या नॉन होमोलॉगस पार्टवर जेवढे जीन्स आहेत ते नेहमी पिढ्यानपिढ्या बरोबरच राहतात आणि बरोबरच पुढच्या पिढीत संक्रमित होतात. यालाच आपण कंप्लीट सेक्स लिंकेज म्हणतो. अँड सेम इज ट्रू अबाउट हियर ऑल्सो. द जीन्स प्रेझेंट ऑन द नॉन होमोलॉगस पार्ट ऑफ Y chromosome also always remain together, inherit together generation to generation. That is complete sex linkage. Why? Because non-homologous parts does not show crossing over. त्यांच्यामध्ये क्रॉसिंग ओव्हर होत नाही. नॉन होमोलॉगस रिजन मध्ये X आणि Y क्रोमोझोमच्या क्रॉसिंग ओव्हर होत नाही. म्हणजेच काय? देयर इज नो चान्स ऑफ genetic reshuffling. There is no chance of genetic recombination, and that's why these genes present on non-homologous parts of X and Y chromosome show complete sex linkage. These non-homologous parts contain the genes and control the different characters. Let me tell you, such as the X-linked traits, say the hemophilia, this disorder, then red-green color blindness, myopia, it is an eye disorder, that is nearsightedness, short-sightedness. These are the X-linked traits, and these traits show the complete sex linkage. There are certain Y-linked characters also, such as hypertrichosis, we shall talk about it later on, as well as ethiosis. These are Y-linked traits and they show the complete sex linkage. Now, incomplete sex linkage. Incomplete sex linkage is exhibited by the genes present on the homologous part of sex chromosome. Incomplete sex linkage is shown by the genes present on homologous parts of sex chromosome. Means what? These genes does not remain together and inherit together generation to generation. They can be separated from each other because the crossing over between the homologous parts of sex chromosome is possible. या ठिकाणी X आणि Y क्रोमोझोमचे हे जे होमोलॉगस पार्ट्स आहेत त्यांच्यामध्ये क्रॉसिंग ओव्हर शक्य आहे. And you know, during crossing over it occurs the mutual exchange of corresponding segments of the chromatids, and so there is an exchange of genes. So the genes present on homologous part can be exchanged with Y and vice versa. So obviously they cannot show the complete sex linkage. This type of sex linkage is called incomplete sex linkage. A very typical example of this incomplete sex linkage, total color blindness. Total color blindness, about which also we shall talk later on. Now, we people have to study about the experiment performed by great geneticist T.H. Morgan for the study of linkage and crossing over. Now, we all are very well aware आता आपल्या सगळ्यांना लिंकेज म्हणजे काय, सेक्स लिंकेज म्हणजे काय, क्रॉसिंग ओव्हर काय हे व्यवस्थित माहिती आहे आणि हे अभ्यासण्यासाठी टीएच मॉर्गनने जो एक्सपेरिमेंट केला तो आता आपल्याला या ठिकाणी स्टडी करायचा आहे. सो लेट अस स्टार्ट द स्टडी ऑफ टीएच मॉर्गन्स एक्सपेरिमेंट. Morgan's experiment showing linkage and recombination. Now all of you are very well known and we have completed the study of linkage and recombination. You know that linkage is nothing but it is the tendency shown by the genes present on the same chromosome to remain together during inheritance, during hereditary transmission. And recombination is nothing but the reshuffling or rearrangement in the structure of chromosome, structure of genetic material, or we can say there is a reshuffling in the arrangement of genes which mostly occur due to the process of crossing over. Now here we people have to study about the Morgan's experiment showing linkage and recombination. T.H. Morgan, he was an American biologist. T.H. Morgan, he was an American biologist and he discovered so many different phenomena such as linkage, sex linkage, crossing over, that is recombination, etc. And he discovered all these phenomena by performing experiments on fruit fly, that is Drosophila melanogaster. Listen, this T.H. Morgan, he was an American biologist and he discovered different phenomena such as the linkage, sex linkage, recombination by means of crossing over by performing experiments on fruit fly. Scientific name Drosophila melanogaster. Drosophila melanogaster. It is a small sized insect. Now question arises, why Morgan selected the Drosophila melanogaster as his experimental material? There are certain reasons. Because this Drosophila melanogaster, this small sized insect, it can be easily cultured in the laboratory. Cultured in the sense it can be easily reared, it can be easily grown and multiplied in laboratory. Laboratory मध्ये, प्रयोगशाळेमध्ये ही जी फ्रूट फ्लाय तिचं संगोपन, जतन, संवर्धन, कल्चर हे सहजासहजी करणे शक्य आहे. Second reason, this Drosophila melanogaster, this fruit fly has a very short lifespan, that is of only 2 weeks. Its lifespan is very short, that is of only 2 weeks, and that's why many generations of these Drosophila can be studied within a very short time. So obviously the inheritance of characters, transmission of characters from generation to generation can be studied within a short time span. अर्थात या ड्रोसोफिला मेलॅनोगॅस्टरचं लाईफ स्पॅन अतिशय शॉर्ट आहे, फक्त 2 आठवड्यांचं. आणि अर्थात त्यामुळे या ड्रोसोफिलामध्ये जे इनहेरिटन्स होतं, गुणधर्मांचं संक्रमण होतं, ते अर्थात अनेक पिढ्यांमध्ये सहजासहजी आणि कमी कालावधीमध्ये अभ्यासलं जाऊ शकतं. अँड मोर ओव्हर दिस ड्रोसोफिला हॅज also a very high rate of reproduction. It means it produces large number of offsprings which are necessary during the experiments, genetic experiments. These are certain reasons why Morgan selected Drosophila melanogaster as experimental material in his genetic research. Now this Morgan performed dihybrid cross experiments. Morgan performed several dihybrid cross experiments as Mendel had performed in garden pea plant. मेंडेलने गार्डन पी प्लांटवर ज्या पद्धतीने डायहायब्रिड क्रॉस एक्सपेरिमेंट्स परफॉर्म केले होते तीच मेथडॉलॉजी वापरून या ठिकाणी मॉर्गनने ड्रोसोफिलावर डायहायब्रिड क्रॉस एक्सपेरिमेंट्स परफॉर्म केले. And as I stated earlier, in Drosophila, the male sex chromosomes are X, Y, and female sex chromosomes are XX, just like the human being mammals. Now, this Morgan also discovered different sex-linked genes, many X-linked genes and X-linked traits in Drosophila. Listen carefully. This Morgan discovered many X-linked genes and X-linked traits, X-linked characters in these Drosophila. या मॉर्गनने या फ्रूट फ्लाय मध्ये ड्रोसोफिला मध्ये असंख्य एक्स लिंक्ड जीन्स शोधून काढले आणि त्या एक्स लिंक्ड जीन्स मार्फत जे विविध गुणधर्म नियंत्रित केले जातात, ज्यांना आपण म्हणतो एक्स लिंक्ड कॅरेक्टर्स, एक्स लिंक्ड ट्रेट्स, त्यांचा आपण शोध लावला. से फॉर एक्झाम्पल, the Morgan discovered different X-linked genes and X-linked traits as first, the color of body. It is an X-linked trait in Drosophila. Second, the color of eye. Third, the shape and size of wings. We know the Drosophila, it is an insect, so obviously it shows two pairs of wings. So I said that the Morgan discovered different X-linked genes, different X-linked traits in fruit fly, such as the color of body, color of eye, shape and size of wings. Among these X-linked traits, among these X-linked characters, let us talk one by one each. First of all, let us consider the color of body. In fruit fly, the normal body color, listen, in fruit fly, the normal body color is brown. Normal body color is brown and controlled by gene Y. Okay? Of course, the Y, it is the wild gene. It is dominant gene. And this Y, normal or wild gene, is dominant over the recessive trait. It is dominant over the recessive trait, that is yellow body color. The gene responsible for it is small y. Just a minute. Here the dominant genes are always represented by symbol plus. Means you must remember that the dominant gene, wild gene, controlling the brown coloration of body is Y plus, and the recessive gene controlling yellow coloration of body is controlled by small y. Color of eye, second character, color of Y. The normal color of eye is red. And the recessive trait is white. White color of eyes is a recessive trait and red color of eyes is dominant, or I will say normal. Now this red color of eyes, this trait is controlled by gene W plus, and white color of eyes, this recessive trait is controlled by simply gene W. Shape and size of wings. The normal shape of size is controlled by gene M plus, small m plus, while the recessive trait, that is miniature wings, is controlled by gene small m. So here actually I have told you the alleles of each other. Now we all are aware all these three are X-linked characters, and all these are that is Y plus Y, W plus W, M plus M, all these are the X-linked genes. Means these genes are present only on the X chromosome, only on the X chromosome. Is that clear? Now we know in fruit fly, as the male sex chromosomes are X and Y, so obviously in male Drosophila only single dosage condition of such X-linked gene is possible, isn't it? As in male Drosophila, sex chromosomes are X and Y, means here only X chromosome is present. So obviously these X-linked genes are present in single dosage. There is no allele on Y chromosome. हे सगळे जे जीन्स मी या ठिकाणी काढलेले हे X लिंक्ड आहे आणि या ठिकाणी ड्रोसोफिला मेल ड्रोसोफिला मध्ये एक X क्रोमोझोम आहे, दुसरा Y, म्हणजे अर्थातच हे जे X लिंक्ड जीन्स आहेत ते सिंगल डोसेज मध्ये असतील. आणि म्हणून हे जे रिसेसिव्ह कॅरेक्टर्स आहेत, ह्या ज्या रिसेसिव्ह ट्रेट्स आहेत, they can express in male individual even if present in single dosage condition. Means suppose these are X and Y chromosomes, that is the sex chromosomes of male, and consider here on this X chromosome, suppose this Y gene is present, recessive gene responsible for yellow coloration of body. So obviously in that male the yellow coloration of body, this trait will be expressed. Why? Because there is no dominant allele on Y chromosome, that is Y plus. अर्थात हा जो रिसेसिव्ह जीन आहे स्मॉल y, त्याला सप्रेस करण्यासाठी, त्याचा इफेक्ट नलिफाय करण्यासाठी दुसरा डॉमिनंट जीन, त्याचा जो होमोलॉगस क्रोमोझोम आहे Y, त्याच्यावर नाहीये. आणि म्हणून मेल्स मध्ये, मेल इंडिव्हिज्युअल्स मध्ये all these recessive genes can express even in single dosage condition, but in females we know there are two X chromosomes, isn't it? There are two X chromosomes, and that's why these recessive genes can express only if present in pair, only if present in pair. अर्थात फिमेल ड्रोसोफिला मध्ये, फिमेल फ्रूट फ्लाय मध्ये हे जे रिसेसिव्ह जीन्स आहेत हे फक्त तेव्हाच एक्सप्रेस होतील, त्यांचे कॅरेक्टर तेव्हाच अवतरित होईल ज्या वेळेला हे रिसेसिव्ह जीन्स डबल डोसेज कंडिशन मध्ये असतील, पेअर्ड असतील. बिकॉज वी नो इफ देयर इज वन रिसेसिव्ह जीन and its allele is dominant, then that dominant allele suppresses the effect of recessive. Is that clear? Now we have to study about the actual dihybrid crosses performed by Morgan. Morgan ने dihybrid crosses कसे केले ते आपल्याला शिकायचे. The Morgan performed several dihybrid crosses to understand the phenomenon of linkage as well as we can say crossing over or recombination. So let us see that crosses. Listen very carefully. Here first of all let us consider the cross A and cross B. In cross A, Morgan crossed double homozygous recessive female with yellow body and white eyes. Yellow body and white eyes with wild male. Wild male, that is the male with dominant character, that is normal characters. You know the brown color of body is dominant character and red color of eyes is dominant character. So he crossed the yellow bodied and white eyed female to the brown bodied and red eyed male. you know, this male is nothing but the wild male, because in this male the both traits whatever considered are normal. Okay, just now we have studied that yellow body, this trait is controlled by gene small y, brown body is controlled by y plus, white eyes, this trait is controlled by W, and red eyes controlled by W plus. Now let me show their genetic constitution, or we can say genotype diagrammatically. We know that in female, as there are two X chromosomes, these are X chromosome, and I am going to show here the genes Y, Y and W, W. Is that clear? I said the female is double recessive homozygous. It means what? Here two characters are considered. One, color of body, second, color of eyes. And the gene for yellow color of body are identical, that is homozygous, as well as the genes for white color of eyes are also identical, that is homozygous. On the other hand, here we can show the genetic constitution of male, wild male in this manner, such as the brown body Y plus. Y plus, it represents the brown body color, and second W plus representing the red eye color. Now when these two parents crossed, initially as usual gamete formation. So here this female is homozygous, double homozygous, so all the gametes form of identical type, that is small y and W, and here the male produces two different types of gametes, half of the gametes with X chromosome containing gene Y plus and W plus, and half of the gametes with Y chromosome. We know there is no question of any such gene on Y chromosome because all the genes are X linked. या ठिकाणी Y chromosome वर हे कुठले जीन्स असण्याचा प्रश्न नाहीये, कारण हे जे जीन्स आपण कन्सिडर केले हे X linked आहे. So the F1 individuals form in this manner. Let me show you. XX, look at the diagram. Y plus Y, W plus W. So the female form with Y plus Y, W plus W. Phenotype, या femaleचं phenotype काय असणार आहे? Obviously here it is double heterozygous condition. ही जी female आहे ती double heterozygous condition दाखवते. याचा अर्थ ह्या ज्या जीन्सच्या दोन जोड्या आहेत त्याच्यामध्ये एक जीन डॉमिनंट दुसरा रिसेसिव्ह आहे, एक जीन डॉमिनंट दुसरा रिसेसिव्ह आहे. याचा अर्थ या फिमेल मध्ये डॉमिनंट जीन्स एक्सप्रेस होतील, अर्थात दिस फिमेल इज विथ brown body and red eyes, that is showing the wild, we can say, characters. And second chance, female gamete when fuses with male gamete, here is the female gamete with small y, W and Y chromosome, XY. Now here the male form is with single Y, single W, and we know that single Y is responsible for yellow body and W for white eyes. So here the male individual is with yellow body and white eyes. Now when the intercrossing between these F1 carried out, when the intercrossing of these F1 individuals is carried out, ज्या वेळेला या F1 individuals intercrossing घडून आलं, at that time the Morgan noticed that about 98.7% of F2 individuals showing parental type of combination of traits. 98.7% of individual with parental type of combination of traits. लक्षात घ्या, या ठिकाणी जेव्हा F1 चं त्यांनी इंटरक्रॉसिंग घडून आलं आणि त्याच्या इंटरक्रॉसिंग मधून जे F2 इंडिव्हिज्युअल्स निर्माण झाले, त्या F2 इंडिव्हिज्युअल्स पैकी जवळपास 98.7% F2 इंडिव्हिज्युअल्स मध्ये पॅरेंटल कॉम्बिनेशन ऑफ ट्रेट्स हे दिसून आलं. म्हणजे काय, पॅरेंट्स मध्ये कॅरेक्टर्सचं जे कॉम्बिनेशन होतं ते इतक्या जास्त प्रमाणात दिसून आलं. याचा अर्थ 98.7% of individuals were either with wide or we can say with yellow body and white eyes, and only 1.3% of F2 individuals showed non-parental type of combination of traits. अतिशय कमी प्रमाणात F2 individuals मध्ये नॉन पॅरेंटल कॉम्बिनेशन ऑफ ट्रेट्स दिसलं. नॉन पॅरेंटल म्हणजे काय, से द इंडिव्हिज्युअल आयदर विथ येलो बॉडी अँड रेड आईज और ब्राउन बॉडी अँड व्हाईट आईज. ट्राय टू अंडरस्टँड. पॅरेंट्स मध्ये कॉम्बिनेशन ट्रेट्सचं कॉम्बिनेशन हे कसं आहे, एका पॅरेंट मध्ये येलो बॉडी आणि व्हाईट आईज आहे, दुसऱ्या पॅरेंट मध्ये ब्राउन बॉडी आणि रेड आईज आहे. हे कॉम्बिनेशन जवळपास 98.7% F2 इंडिव्हिज्युअल्स मध्ये आढळून आलं आणि फक्त 1.3% इंडिव्हिज्युअल्स मध्ये नॉन पॅरेंटल. नॉन पॅरेंटल म्हणजे पॅरेंट्सच्या व्यतिरिक्त दुसरं कॉम्बिनेशन दिसलं, सच ॲज द इंडिव्हिज्युअल विथ येलो बॉडी अँड रेड आईज, ब्राउन बॉडी अँड व्हाईट आईज. On the basis of this result, he explained that the parental combination is due to the linkage. त्याने असं सांगितलं, हे जे पॅरेंटल कॉम्बिनेशन जास्त प्रमाणात दिसतंय ते कशामुळे दिसतंय, लिंकेजमुळे. आणि जे नॉन पॅरेंटल कॉम्बिनेशन आलेलं आहे that is due to the recombination, means due to the crossing over. Also, he performed one another cross. Let us talk about that cross. हा दुसरा क्रॉस कसा आहे ते आता आपण या ठिकाणी बघूया. In second cross, Morgan crossed the double homozygous recessive female with white eyes and miniature wings. You know, white eyes and miniature wings, these are recessive traits. White eyes, this trait is controlled by gene W, and miniature wings by small m. And this double homozygous recessive female crossed with wild male. Wild male means the male in which both the traits are dominant. That is, with, we can say, red eyes and normal wings. We know that the gene responsible to control the red eye color is W plus, and the gene responsible for normal wing is M plus. Now let me show here their genetic constitution, genotype, diagrammatically. Suppose in this double homozygous recessive female, I am going to show here is a pair of gene WW controlling character white eye color and MM controlling the character of miniature wings. Why the male with XY has single dosage condition the gene for red eye color is W plus and gene for normal wings M plus. When the crossing is carried out, as usual, the gametes produced by this double homozygous recessive female are identical, that is all gametes with W and M, and here the male produces two types of gametes, half of the gametes with X chromosome and gene W plus, M plus, and half of the gametes with Y chromosome. So, after their fertilization, the offspring produced are like this. That is, the female with W plus, M plus, and WM. Okay? Means here, the female individual has double heterozygous condition. It is double heterozygous condition. आपल्याला कळत असेल W plus W M plus M double heterozygous condition, and its phenotype is red eyes and normal wings. Why? When the X chromosome, that is one female gamete, unites with another male gamete with Y chromosome, then its genotype is WM. There is no question of allele. Okay? So here, this male individual is with white eyes and miniature wings. Now, when the intercrossing of these F1 individuals is carried out, when the intercrossing of these F1 individuals is carried out, Morgan noticed that, listen, Morgan noticed that in F2 individuals, about 62.8% of individuals with parental type of combination of traits. Parental type of combination of traits. याचा अर्थ या F1 मध्ये इंटरक्रॉसिंग केल्यानंतर जे F2 इंडिव्हिज्युअल्स निर्माण झाले, पुढच्या जनरेशन मधले इंडिव्हिज्युअल्स निर्माण झाले, त्यापैकी जवळपास 62.8% इंडिव्हिज्युअल्स मध्ये पॅरेंटल टाइपचं ट्रेटचं कॉम्बिनेशन दिसलं आणि फक्त 37.2% इंडिव्हिज्युअल्स मध्ये नॉन पॅरेंटल किंवा न्यू कॉम्बिनेशन ऑफ कॅरेक्टर इज सीन. ऑफ कोर्स ऑलरेडी वी हॅव सीन in this cross, in cross A, F2 individuals showed 98% of 7 parental type and 1.3% non-parental. In second cross, he noticed 62.8% parental type and 37.2% non-parental type. From this, he concluded that the parental combination of traits in F2 generation is due to linkage. Listen carefully. या दोन्ही क्रॉसेस वरून त्याने असं सांगितलं की F2 इंडिव्हिज्युअल्स मध्ये पॅरेंटल टाइप ऑफ कॉम्बिनेशन ऑफ ट्रेट्स जे दिसतं, पॅरेंट सारख्याच गुणधर्म ज्या F2 इंडिव्हिज्युअल्स मध्ये दिसतात ते लिंकेज मुळे. म्हणजेच काय? The ability of genes present on same chromosome to remain together and inherit together. म्हणजे X क्रोमोझोम वर असणारे जे जीन्स आहेत ते सोबत राहण्याचं आणि पुढच्या पिढीमध्ये सुद्धा सोबतच संक्रमित होण्याची जी त्यांची टेंडन्सी आहे, त्यामुळे F2 इंडिव्हिज्युअल्स मध्ये अशा प्रकारचं पॅरेंटल कॉम्बिनेशन दिसतं. आणि जे नॉन पॅरेंटल म्हणजे पॅरेंट्स पेक्षा वेगळ्या गुणधर्मांचं जे कॉम्बिनेशन दिसतंय, दॅट इज ड्यू टू द क्रॉसिंग ओव्हर और दॅट इज ड्यू टू वी कॅन से रिकॉम्बिनेशन. अर्थात जे न्यू कॉम्बिनेशन्स, नवीन गुणधर्मांचं कॉम्बिनेशन ज्या F2 इंडिव्हिज्युअल्स मध्ये दिसतंय ते अर्थातच रिकॉम्बिनेशन मुळे किंवा क्रॉसिंग ओव्हर मुळे असतं. मोर ओव्हर त्याच्या पुढे जाऊन त्यांनी असं सांगितलं, द जीन्स which are tightly linked on the same chromosome. Listen carefully. the genes which are tightly linked on the same chromosome, means closely placed on the same chromosome, have less chance of recombination. Listen, अर्थात एका क्रोमोझोमवर असणारे हे जे लिंक्ड जीन्स आहेत ते जर क्लोजली प्लेस्ड असतील म्हणजे स्ट्रॉंगली लिंक्ड असतील तर अर्थातच त्यांच्यामध्ये रिकॉम्बिनेशन होण्याचे चान्सेस कमी आहे आणि आपल्याला या ठिकाणी दिसेल. लुक हियर, purposefully I have shown here on this X chromosome I have shown these two genes placed quite close to each other. Means here in this example YW or this Y plus W plus, these genes are closely placed, strongly linked, and that's why in F2 generation the chances of non-parental or recombinants is less. On the other hand, here in this condition, please notice, या ठिकाणी आपल्या लक्षात येईल की W plus M plus, these genes are present on same chromosome, but they are not strongly linked. Just like YW, these are not strongly linked. Means these are placed on same chromosome, but apart from each other, away from each other. They are loosely linked, and that's why such loosely linked genes have more chances of recombination, and that's why in this cross the non-parental or recombinants appeared about 37.2%. From this experiment, Morgan concluded that the genes present on same chromosome has ability to remain together and inherit together in successive generations. It is called as linkage. When this linkage is seen in sex-linked genes, then it is known as the sex linkage, and the chances of recombination is more in those genes which are loosely linked, and chances of recombination is less in strongly linked genes. Here we have completed this Morgan's experiment showing linkage, sex linkage and recombination. So let us stop here. Thank you. Bye.